Okay. Morning, everyone. Can everyone hear me clearly from the back? Good, because then I'm gonna move this microphone away because I'm really bad at staying in one place. So I'm kind of twitchy. Although not usually in the morning, but I'm awake. So thank you all for being here. I'm Nicholas Chia. I work at the Mayo Clinic. A little background on me: I got my PhD in physics at Ohio State University. I moved on to University of Illinois, where I worked on origin of life research, actually, and early life research with Nigel Goldenfeld, Carl Woese. Then I somehow went to the Systems Biology Institute for like a little bit, and then I came to Mayo Clinic because they said, well, we want to start a microbiome program, and are you in? And they were very convincing, so I joined and started using ecological knowledge to really look at medicine. So just a little bit of background on the problem I study. I study colorectal cancer. This is a problem that Ron has already very nicely presented. The only things you need to know is that colorectal cancer is bad. It is 85% sporadic, which means environmental, which is not to say there's not host genetics involved somewhere in the 85%, but we don't know it. And so, it's an environmental cause. Environment, in my mind, is really equivalent to the microbiome. And I'm gonna show you a couple techniques for looking at the microbiome as a set of interactions, specifically metabolic interactions. And so one of the things I thought that would be helpful as I was watching the other talks was to go through the assumptions underlying the work. And one of the assumptions here is that I can capture microbe-microbe interactions through their metabolic cross-feedings. And that this modeling has no explicit regulatory elements or signaling elements in it, but it's working under this assumption that we can assume some kind of coevolution, right? That microorganisms have sort of adapted to each other, and that I'll show you that this. Well, I can show you examples where this works pretty well. There were a couple of things here that I thought I would just run through quickly before getting to the techniques. One is that little domain familiarity with the information. I always think it's helpful to touch the data. I always tell people, touch the data, open the raw file. I think everybody thinks I'm a little crazy because, of course, it's a huge text file. Why would you look at it? But it gives you familiarity with the data. So in this field, one of the issues with colorectal cancer signals and microbiome data is that you can't quite get consistency. There have been a number of meta-analyses out, actually even just this year. This heat map here is from Pat Schloss, where if you had consistency between these different studies, you would have all red or all blue in a row. And as you can see, there's always some pain in the butt somewhere that won't agree with everyone else. And that's a problem because it's not just saying, some studies catch a signal and some studies don't. It's saying some studies say, porphyromonas cause cancer, and one study says, nope, it's actually protective. And so that's kind of an ongoing problem in the field. One of the reasons I think that occurs is because the underlying distribution we use to assess these associations is wrong. So I can show you many examples of low abundant taxa, which are often the same ones that are considered putatively related to colorectal cancer, that follow something closer to a power law than any kind of symmetric Poisson Gaussian normal distribution. And so what that means is that then when you assess things with a Gaussian distribution, things become very sensitive to outliers, right? And very often you could see, if you plot the data the right way, you can see that a lot of these signals are being driven by outliers in the data set. And so that's something where a little familiarity helps, because it's a little different than human data, I'm noticing. You guys have much nicer distributions, I guess. It sounded like. But in the microbiome, this is what it looks like. The other thing is you have to think about disease-specific factors. So you have to recognize your disease. In this case, what I'm going to put forward is that colorectal cancer isn't really one disease when you look genetically. There are multiple lineages of colorectal cancer, and they're each progressing in their own way. And so why would we expect them to have the same microbial influences? We don't expect them to follow the same lineage. And so in colorectal cancer, two of the main paths are deficient mismatch repair and proficient mismatch repair. Mismatch repair for the microbiologists is like homologous recombination. It's your way of repairing things, and for the human geneticists, it's mismatch repair, I guess. I don't really know. But basically it corrects errors, and so when you don't have it working, you pick up a lot of mutations and you become this hyper mutated type. And two of the starting mutations, down the typical lineage of 1, you would expect APC, KRAS, so on, and these kinds of things, but you almost never see that on the other side. Instead, you see a BRAF V600E mutation, and you see a different set of mutations in that progression. So to convince you that you have to take the biology into account, I just put this up really quick. This is calculating the amount of variation that comes from each variable in our data set. And this is order dependent, so it's a method that looks for how much variation can be explained by one of these factors like BMI, and then from that matrix, which will have the OTUs and the samples, it will then subtract out that variation and give you a residual matrix, and then you can do your next calculation of variation on it, and you keep going down, and you can see even accounting for everything else we have on top, when we get to MMR status, it's still the most explanative thing. So the other way to think about this is that if I flip this up to the top, the MMR bar would be huge. And this is largely because MMR status is in fact is confounded by these other factors. So particularly age and the sample location. DMMR tends to be much more proximal, and PMMR tends to be much more rectal. So, oh yeah, a mismatch repair. Yeah, so that's the homologous recombination part, where if you have, if it's working, you can repair your genome. If you have some kind of lesion or something like that. If it's not working, any kind of break in the DNA leads to errors, and so you get hypermutated. And so that's sort of two different paths towards cancer. So just to show you that it makes quite a difference, I plotted a Venn diagram of the tumor associated microbes in DMMR and the normal associated microbes in DMMR, and I did that for PMMR too. And really all you need to take away from this, to not get too complicated, the only thing you need to take away from this is that there's not really a lot of overlap between those two. They really are looking like separate diseases. So deficient mismatch repair as opposed to proficient mismatch repair. So deficient mismatch repair is when the MMR, the mismatch repair genes, are not working, and proficient is when they are working. So for the purpose of this talk, it's not so important, but just to show you that there are two different disease subtypes here, and they need to be considered separately. And so, the question of, can we apply this principle of subsetting the disease, just like we have in other areas of colorectal cancer, can we apply that to the microbiome? The answer is yes. Now, just to convince you, or just to show you a little bit of a highlight for those of you who do microbiome research, or those of you who are sort of curious about it, I've listed now some of the main signatures from the deficient mismatch repair side, but when we look at on and off tumor, and the proficient mismatch repair side. And some of the things I would just bring to your attention right away are on the deficient side. So for those of you how many of you are microbiome researchers versus how many? And then how many human? Oh boy, okay, I'm not very human. So I'll try to, I don't usually give talks to human people. Let's see. So backing off a little bit, there are a number of main signatures in the literature for colorectal cancer and the microbiome. Many of them are like bFrag, for instance, up here. And when people do their analyses, they take all colorectal cancer at once, and they either take stool or tissue. And so for instance, bFrag has been found to be in proximal biofilms by Cynthia Sears's group. And we're interestingly finding it very strongly associated with the deficient regime, which is also the type that is proximal or more proximal, and we don't find it in the proficient mismatch repair side. So this is sort of telling us that there's a signal that people are seeing that really only applies to 15% of the patients, because deficient mismatch repair is a subtype that only affects 15% of colorectal cancer patients. And the same with many of the other signals. So like Fusobacterium is a classic one. I think that's probably one of the biggest stories in colorectal cancer. And again, if you look at this cohort of 15% of the colorectal cancer cases, you have a p-value that jumps down below e to 10 to the negative 200. Whereas when you put your group together, it's like, the p-values 10 to the negative 20 or so. So it's telling you that you get a much better signal when you split things up according to their biology. So this is, the whole point of this is just to tell you to keep track of your problem. And the other signals are very much the same way. They recapitulate what others have found, but with better p-values and splitting things when you split things apart the disease-wise.
So is it 16S analysis?
Oh yes, this is 16S.
And how many samples? Because the p-values after FDR are really low.
Yeah, so the number of subjects in this study are 83. I took out my sample description slide because I want to try to highway us to the tutorial, but these are good questions. So good scientific questions. So 83 people, the sample number is something over 600 samples. Because what we did was, so I work in, the other funny story is I work in Department of Surgery. For some reason they decided they needed a physicist in Department of Surgery. They've made a bunch of bad decisions. They also thought they needed a physicist on their tumor board. Yeah, I don't know what they're thinking. But anyway, so what these surgeons very nicely do for me is they go, oh, you want colons? I can get those. And I said, okay. And so they're like, yeah, we take them out. Like, what's the big deal? And so the pathologists thought this was a big deal, by the way. They were not so much of a fan of giving me the colons. They were kind of like, we need those. The surgeon's like, we don't do anything with this. And I even had surgeons who were like, oh, I could rip out feet of stuff and nobody would know the difference. So they're anyway, they don't think much about it. But anyway, what it allowed me to do is it allowed me from these colectomies to sample location-wise, and then sample on the tumor, off the tumor. And so that's actually how I could get the location effect for these because I had multiple samples of each. So yeah, it was quite a lot of sampling. And then we took scrapes we took scrapes and we took tissue. So with the scrapes, we were hoping to get a little bit more of the mucosal surface, and with the tissue, and if there was stool, we tried to take stool.
And when you say normal, this means a normal tissue from
adjacent, yeah.
To the tumor.
Yeah, and this is the case where, of course, you would like to have normal, but I'm told they don't remove colons from normal people. You laugh, but actually, we did a study. We did a study in endometrial cancer, and we had one reviewer that insisted we have normal controls. And we tried to describe to this reviewer that one does not perform a hysterectomy on normal, healthy women, and they were very confused by this. So it happens. Huh? Yeah, so that is actually a good idea. So those are, so you can't get normal, but you can get differences, right? And you can also get IBD, you can get fibroids. Yeah, it's like not perfect, but you can do it. We also can grab from colonoscopy, you can grab bites of normal tissue, like through their screening colonoscopy, although surgery turns out to be different from colonoscopy because it's even though it's the same bowel prep and everything, it's a completely different procedure. So just the act of surgery is this whole thing that changes your milieu. So how can we tell that the two disease paradigm is better? It's because we have better statistical power, even though we cut our cohort down in size. So when you cut your cohort down in size and the signal goes up, that's a good sign. Okay, so now I'm going to tell you about how we looked at some of the community characteristics of these microbes. So that was just background. So first, one of the questions we had starting in the research was to look at H2S production, right? A lot of people look at different metabolite productions. You can use it to look for anything. Our particular question was focused on hydrogen sulfide, which is a genotoxic agent. But it's also an important signaling agent, and it's made endogenously by the gut. But in high amounts, in theory, it would cause DNA damage. And we thought this is a lot like UV rays, right? You go out in the sun, it's fine. You go out in the sun too much, and you start breaking down DNA, getting mutations, and you get skin cancer. We kind of thought like the sun doesn't shine, but we thought, you know, you make chemicals that cause DNA breaks. It's kind of going to be the same thing. But however, this is a kind of a hard thing to assess because H2S is a gas that is very volatile. It sort of escapes very readily. Unless you're going to like sit there and try to measure this as like the poop is coming out, this is going to be like really difficult because as soon as it's out, you know, the gas is going to be leaving the system. So we wanted to use metabolic modeling to try to help us assess this, and also because it seemed like more fun than trying to figure out how to measure fresh poop. So there are a couple steps here. First, you have to take your 16S profile, and then you have to use those 16S profiles to identify a bunch of metabolic models that you want to bring into your community metabolic model. Then after that, there's really just there's a big step where you have to get the models to talk to each other because not everything is really standardized in terms of biochemistry in the field. So you have to bring all the models together, and then you put them together, and you can model what's going on. And we used a tool called Mecom that's developed by some of our collaborators, and then we can assess the results of that modeling and ask how much H2S is being exported out of that model. So this is not quite up to date. This is just to say we have a pipeline for looking at 16S operations. We glued this together in the early days, and probably only part that's still relevant to this is that we use V3 and V5, which sometimes do not merge completely, so they don't overlap in the middle. You don't get quite enough coverage. They're a little bit further away from each other, so you can get this gap in the middle. And so we do just a couple of things to make sure we can still analyze that well using both sides. And then the rest of it, I think we've pretty much replaced with like Data2 and other things by now. But the goal is ultimately to get out a profile, and this plot over here is showing what happens, that you get different results under different OTU picking circumstances, and it matters whether you use read 1, read 2, or you use both. And then we also compared to closed reference. So we use both when we can, and then you get some kind of taxa profile out. This step is more important than it sounds. It's not in theory very hard, but in order to map to metabolic models, you first have to map to genomes. And in order to do that, you create a 16S database based on the model set that you have. So out of Patrick and Agora, you download those genome sequences, you take the 16S, you sort of, mock grab the region that would, be amplified by your primers. You make a database of that, you keep the mapping to the genome, and then you start looking at your OTUs, sequences, and what would match in the 16S database. You have some kind of identity cutoff where you say, well, if I can't get anything nearby, I don't consider that as something I can model. And then, and it's otherwise kind of like a blast search. We use vSearch. Otherwise, you look at best alignment based on score, but then of course 16S is not really like the fastest moving marker in the world, so you have some ties. So you first looked at the best score, and then you can look at the highest quality genome, because if you're going to be tied anyway, you might as well take the metabolic model that's like nicer. And then if tied, we try to grab the type strain, which usually has the best model as well. So then you download the model corresponding to that 16S sequence, and you put together your whole community. Wait, I'm sorry. That's 70% most of the time. If we count by, about 70%, and at a little bit more of a stringent threshold than this. I think if we go down to 90%, I forget what it is, but it's around there. And also, we've been adding genomes into the database that sort of help us get a little better coverage. So, or your alternate path when you don't have a mapped, you don't have a metabolic model already built is we worked a long time on automated metabolic model building. This is actually kind of a frustrating problem because you can get annotations really easily. You can try to annotate your genome and then figure out what reactions belong, but you're always missing a set. And the classic way of doing things that Bernard Paulson introduced is to kind of go through and do this by hand and figure out like what your missing reactions are, right? But this is actually a pain in the neck because for the sake of like, the 20 reactions. Oh, sorry. A metabolic model is a list of all the chemical equations that occur, the chemical reactions that would occur inside of the cell. So this largely came out of the chemical engineering field, and one of the things they do in the chemical engineering field is they worry about bioreactors, and they say, if I'm working for an oil company, how do I refine things, and how do I sort of split off the byproducts of my first reactor into the second, third, fourth, fifth reactor? And so they really model this by coupling a bunch of chemical equations together and asking, how do I optimize the output? And Bernard Paulson's insight was that you could do the same thing with E. coli, right? You can consider it a bunch of reactions, and you could say, how do I put this together so that I can model it as a bioreactor? So so you end up with a huge stoichiometric matrix where basically you've encoded the metabolites on one side and the reactions on the other. And so if you have a reaction A goes to B, you have in the A column a -1 and you have in the B column a +1. And so it's a conversion. And you do that for all of them. But the problem with a metabolic model is the problem and the strength of it is that it's very precise. And so if you're missing a link, your whole model doesn't work, right? So you have to do something about that. And what people do is they basically, for the 1% of the model or the 5% of the model that they don't have any information on, they have to figure this. In the past, first they had to figure this out by hand. I think maybe 10 years ago, there was a paper out by Paulson and Thiele saying this takes about 6 months for a single microbe. And of course, for microbiome work, that's sort of unacceptable because we have, as you've heard, a lot of microbes. So automated metabolic model building is one of the ways we can address this. And there's a paper on this that is already there. And this is just an ROC curve that shows how well we do. It should be true and then false. When we compare to a hand-curated model, two hand-curated models, and this is an ROC curve based on our likelihood score. So the more likely we consider a reaction to be in the model, the more likely it is to actually be in the model, and we can use that to more accurately include reactions. And I luckily don't have to talk too much about it because whatever interface we have is in K-base or Patrick. And for the most part, now you can just go there and grab those models. Yeah.
Besides the sequences, what other data do you use?
GCMS.
Are you using any metabolic data?
Oh, yeah, we do for validation. So I'll show that later.
So you don't include any metabolic, so you don't know what is the actual metabolic product of the bacteria that you're looking at your sequencing. You're just assuming a model
Yes, yes, and and so in theory, because it's a likelihood model, we actually could inform the weights of the metabolite nodes using the metabolomics, but we don't in this case because we wanted to show that it works first. Also, there are some other technical challenges because metabolomics gives you a concentration, and the result of metabolic model is usually to run a flux balance analysis, which is to say it asks how much flux is being produced at a given time, which doesn't necessarily correlate to accumulated concentration. Yeah, one is kind of like the motion, and the other is the amount. So there are a couple of things there, and then there's the fact that I think only about a third of what you get out of metabolomics actually corresponds to metabolites that appear in the model. So a lot of the metabolite signatures you get aren't things that you can assess. There's some issues. We're working on it. Other people are working on it too. So there's a bioRxiv paper describing Mecom, so I don't have to describe it in as much detail. And this is a tool that works with Cobra Pi, and so you can go there and read it, but that's someone else's work, so I'm not going to give you a tutorial on that. I'll just say that one of our most popular tools, which I did not think would be super popular, we wrote this internally for ourselves, has actually been called Mackinac. One of the people in my lab from Wisconsin wrote this. Mackinac is one of the longest bridges in Wisconsin. It's this gigantic bridge. I've never been on it. I've heard it's really large. It's just supposed to get you from like one person's version of a model to someone else's version of a model, right? So I always think this is kind of like the XKCD comic where somebody gets up and goes, there are 10 different standards in the field, this is ridiculous. And then the next thing that happens is now there are 11 different standards in the field. And so this is kind of what happens. And I said, I don't think this is the right approach to just keep trying to build more standards, because what happens is we just end up with even more of a problem. Instead, I sort of took what I call the Google Translate approach, which is instead of trying to build a dictionary that's going to work for everything, build a translator. And so Mackinac is the translator, and oddly enough, I think this is our most downloaded tool because I guess it's a very frustrating problem to have. So what we did, and so this probably gets at your question about the metabolomics. So what we did to assess hydrogen sulfide is we recognized that the hydrogen sulfide was coming out of, for Fusobacterium and other sulfidogenic bacteria in our systems, was coming out of the amino acid pathway, and that there were certain markers in the amino acid pathway that were more specific to the microbe. So this way we could assess the microbial production of H2S. So we then use the amino acids as our surrogate for looking at H2S production because they're part of this reaction. And so what we're showing here is a diagram of just the pathway, and then on this side, this is our actual metabolomics. These are our metabolomics results. And in red is the DMMR side, solid is tumor, striped is normal. And you can see that we were thinking that there's going to be more H2S in the tumors and from microbially produced H2S in the tumors, and from what we show in the data, that's what we're seeing. So that's the result of us performing this modeling. So Mecom has a way of did I get this in the right order? So I forgot to put the other slide in. The last. Let me just go back real quick. So after we build the community model, we run Mecom in a mode that makes sort of a steady state assumption and says, okay, if this is the configuration of the microbiome, what is the metabolism you have to keep that configuration? And based on that, you can sort of assess what the output of that metabolic network would be. And in our case, we found that there was higher H2S, both in what we modeled and what we show here. And we can show that we have higher H2S, and that's validated. So I thought I'd stop here for some reason. Any questions on that part? So that's one type of modeling, and I want you to lock that in your mind on one side. That's a type of modeling where we took the whole community at once and we modeled the whole thing, and we didn't really consider microbe-microbe interactions except sort of except in terms of taking into account when we model the whole community, but we don't focus on the microbe-microbe interactions and asking what are the key species in that community, what are the key ecologies. So I'm gonna give you a separate analysis now that's similar, but I don't want you to get confused between the two, because this one now, instead of assuming the whole community is growing at once, it's treating each microbe separately, which in my view is reflects what a microbe does, right? I mean, they're not sort of, they're not an organized, they self-organize. They don't really, have a decision like we're gonna have, 10 secretaries to every professor or something like that, right? So they're growing independently, and they have their own metabolic influence, and they have their independent behavior. So here, what we were trying to do is figure out who are the main influencers of the microbial community by looking at the microbe-microbe interactions. And in this sense, we're really trying to find the keystone species for each group of either for cancer or for non-cancer. So as a quick overview, there's this nice Nature Communications paper that was really put together by Jae-Yoon Sung, who's actually now an assistant professor at Mayo. This is the final diagram he came to in the paper. As you can see, there's a lot of detail and there are a lot of connections here. I'm going to try to break this down for you because I was involved in this. I have a little bit of the history and can maybe make this huge diagram easier to understand. I always think these summary diagrams are very nice, but it's actually a little bit hard to read, right? I mean, you can see a couple of trends, but you can't necessarily see what's going on. So I have to start with the story of how this paper started, which was when Jay became a professor. When he became postdoc, he was working in Korea, and he said to me, well, there's no microbiome research there, and I want to start some. And he said, I want to put together the network, the metabolic network of microbe-microbe interactions. And I said, good luck. I said, the problem with that is that we don't really know how the microbes, what microbes are using which metabolites. He said, well, why don't I use the transporter database? I said, well, the transporter database sucks, so good luck. You're never going to get it. So anyway, a year and a half later, Jane came back to me and said, I read 200 plus papers and I re-annotated the whole thing. And so he characterized a huge number of metabolites and microbes, and this was amazing. And so out of that, he then built the microbe compound bipartite network. And what that means is that for each microbe, he asked which metabolites do they take in and which metabolites do they excrete. And yellow is an import, blue is an export. So export means that microbe makes that metabolite and spits it out. Import means it uses it and eats it. For the bottom part shows the different metabolites and roughly separated by category. And the top part are the different microbes. And you could see that there's a little bit of structure here in terms of, certain microbes break down things and then other things use them. And so there's interesting cross-feeding going on. And then once you have this microbe metabolite relationship, you can ask, what are the microbe-microbe relationships?
He did it by hand,
Yeah.
looking at.
Yeah, he's insane. Yeah. He has it in a spreadsheet. I'm on the paper. I can pass you the spreadsheet. It's insanity. Sorry, I'm hard of hearing. Not really, but I'm shouting up here, and for some reason I've oh, sorry, wrong way. Also. Oh, red. Red is macromolecule breakdown. So it's not really an import, but it's not really an import, but it's kind of equivalent to importing it and then breaking it down and spitting it out. But it's not really. You instead that organism secretes an enzyme that breaks something down outside of its wall, and then it ends up being shared with other microbes. So it's basically a lot like a yellow line, but with a technical difference.
So the dots here are the microbes?
Yeah, the dot.
So, yeah, where's the graph? Where are the two parts of the graph?
Well, the bottom part of the metabolites.
Okay, okay.
Yeah, so the metabolites on the bottom are the circles, the white circles, and then the top, the colored bits are the microbes. Yeah. It's a lot of interactions. He's a little bit OCD.
Yeah.
His office is much neater than mine. But on the other hand, if you ever want to drive him crazy, just like move things in his office. Like just start shuffling things. Not that I do this at all. So anyway, so then he put this together, and this is how he kind of got there. And you can see, with the goblet cell, it secretes mucin. It has a bunch of relationships with key bacterial groups, and the different microbes, the different classes of microbes interact with each other. And the purple lines are sharing in terms of, I produce something, and then you eat it, right? And so there's this cooperativity going on in that sense. And then you can also compete as well. So he sort of can just put these interactions together. And the interesting thing he did with this is he said, well, now that I have this, how can I calculate the influence? Well, and this comes with an assumption that the microbe that is most influential is the one that makes a metabolite or a public good that everybody else wants, right? It's like if I had if I had a computational tool and everybody used it for their research, I would be, the most influential person. This is not true, but I would be in some sense the most influential person because everybody would use my tool, right? And this is what we're asking for the microbes. If you make something and a lot of different microbes use it, then you're kind of a keystone species. And basically, what's shown in the equation is a way of counting the number of dependencies weighted by the abundance of that organism. And so what he showed in diabetes, because he was working with public data sets, he grabbed this cohort of diabetic patients and non-diabetic patients, type 2 diabetes, and yellow is diabetic and blue is normal. And he pulled out the normal associated and the diabetes associated microbes. And he could show that there are a number of microbes that are not really considered to be diabetes associated, like Akkermansia, mucinophilia, often this, but it has a huge amount of influence supporting the other microbes that are considered type 2 diabetes microbes. It's up there. It's up there, and so what's interesting is just because a microbe, and a little bit of background on that microbe, it's considered healthy in many contexts, and so it's considered one of the sort of primary microbes that sort of then breaks down mucins and supports the rest of the community. But what you can see is that even though it's a healthy microbe and it's kind of confusing why it's associated with type 2 diabetes, it may just be that it's supporting the rest of the type 2 diabetes community. And so rather than having a direct interaction, you can also ask who are sort of the keystone species supporting a bad microbiome or who's supporting a normal microbiome. And so it doesn't have to be a direct interaction for this to work. And then also out of this, you can start asking other questions. You could start asking what are some of the different supportive metabolites on one side versus the other. And so you can see, on one side there's sort of more cellulose, hemicellulose, and the other more starches. And so you can ask yourself, how do I modify my diet to do this? So we wanted to do the same thing, and we really wanted to broaden this. And but I didn't want to do it Jay's way because that sounded painful. So I decided we would take advantage of our metabolic model building in order to do this. And the difference between what we're doing and what Jay did is Jay literally counted for every single microbe, how many cross feedings there are possibly. We took each metabolic model and built a pairwise model and asked, well, how much do they affect each other's growth? How much do they grow by themselves? How much do they grow together in some environment? And then we use that as our for our influence scoring. And out of that, we assessed a very similar quantity, but based on the interactions we calculated in that environment. So in order to do this, we used a tool named MMinte. Oh.
Isolates for that to assess the microbes alone versus.
Oh, sorry, we did that computationally. Okay. Computationally.
Looking at again the 16SO2.
Yeah, I'm sorry. I should really preface this by saying I really am not qualified to do any experiments. I have done experiments in the past. I am actually technically in charge of the lab right now because my colleague left and left me in charge of her lab. I asked them to order a flamethrower recently because I wanted to see if they would do it. The answer so far has been no, but that's about my lab experience. So I don't really have anything else I do. But we do this computationally. So the goal is really to assess the interactions computationally, because if you had to do it experimentally, you could, in theory, do it, but it would be potentially laborious. The different microbes grow under different culture conditions, and really you want them all under the gut condition, which is kind of hard to replicate. So
in that case, how do you differentiate between the effect of one microbe versus an interaction between a pair?
One.
Maybe I didn't understand.
So we modeled the growth of that microbe alone. So we put it in an environment. We say, what is the growth rate of this microbe by itself in that model?
How do we know that? That's exactly what I'm asking. If
Oh.
For example, so you assume a growth rate model, and then.
Oh, no. So based on the metabolic. Oh, I see. I skipped some steps. Okay. So flux balance analysis does this nice trick where it says, in this environment, assuming the organism is optimizing its growth, what are the fluxes through its reaction network? And growth is captured by a biomass reaction, which basically is a bunch of DNA, protein, lipids that would make up a second cell. And so by asking what is the maximum rate at which that network can produce biomass, you have a surrogate for growth rate. These are really good questions. This sort of saves me from screwing up. So this is really good. So I actually, I wish other people should ask more questions. But it's yeah. So that's how we assess growth, by looking at the flux into biomass. So it gives us a rate of biomass production, and that can be that output can be parameterized to the growth rate for that organism. And so we could do that with any organism. But then we can also put two models together and put all the reactions they have together into separate compartments, and then do the same exact exercise and ask, now what is the biomass production of this versus this? And if this microbe is making something that is really, really useful for this microbe, then its growth rate will go up, and vice versa, right? If this one is eating up all the metabolites that this one needs, then then both their growth rates will go down because they're competing for the same metabolites. Yeah, so that's the basis of it. So the other way to think about it in a metabolic model is to think about rate limiting metabolites, and so you're really usually in your model constrained by whatever your rate limiting nutrient is. And so you're asking yourself, when I put two microbes together, do they get past a rate limiting nutrient because they have some kind of cross feeding that mutually is beneficial, or are they competing for a rate limiting nutrient and both of their growth rates are dampening down? And that's what we're assessing out of by putting these models together. But anyway, I'll go through the steps of MMinte. MMinte is made up of 7 widgets that do exactly what I just described, but in an automated way. So first, it grabs the representative OTU table, which you can grab out of Chime or Data or whatever, and it builds working representative O2s based on the sequence. It takes those sequences, it gets a genome ID out, so it carries out some of these steps we were talking about before. It then grabs those IDs and then grabs the models. At this step, I just want to point out one thing. We get people who try to use MMinte, and I know pretty much whenever anyone tries to use MMinte, because this was originally slaved off of our automated metabolic model building algorithm in KBase, which is a DOE resource for metabolic models and other bioinformatics tools. That is like often broken, so the models are not always there, and there's not much we can do about it. So our algorithm tries to grab those models. By default mode, it tries to go there to grab the models and just give them to you so that you don't have to bother. But very often this breaks, and we get an email saying, well, everything broke on widget 3. And there's not much we could do. However, there are other resources. For instance, you can download Patrick models, and you can download models from the Virtual Metabolic Human, which is a resource set up by Ines Thiele that has a collection of models, the Agora models. So there are other resources that have metabolic models already pre-built that you can download from and use.
Metabolic models exist from how many species?
I think Agora has about 800. Patrick has a few thousand, like 2,000 or something. And of course, though, we wanted to use the automated metabolic model because there are more, genomes than models. But if you grab the existing databases and just go to the closest, that I think is probably the best you can do right now. Oh, okay. So then you grab those models, and you can build 2 species models and assess them alone and together. And what you're looking for is, do they grow faster together or do they grow slower together? And that sort of gives you, for each interaction, that gives you whether you have a positive or negative interaction between those 2 microbes. And then you have the growth rate predictions, and then out of those growth rate predictions, you build an interaction network. So this paper is already out, and you can access the software and go to the GitHub. Using Mike Mundy's GitHub over here is the newest one, and if you go to the old one, it'll tell you to go here. If you go to the README file, it's very easy to install. You can just pip install it, and what you get out of it at the end is you get a table of interaction values. So you have in the V column, you'll end up with the growth rate when they're together. In X, you'll end up with the growth rate when that organism is alone. And what you can see is, for the first interaction, for instance, you have mutualism because together, alone, they grow very slowly, right? The biomass production rate for the first organism, which is this Clostridium, and the second organism, which is this E. coli, are very low, 0 and 2. But then when you put them together, they grow really rapidly because there's some kind of cross-feeding event that allows both of them to grow in that environment. And you can do this for all the different microbes. And then what you do is you basically score the influence. I do this by spreadsheet. This is terrible. My postdoc hates this. But I do it by spreadsheet because I find it easier. And I'm kind of too lazy to make like a program for everyone, but and it's not that hard on the spreadsheet. So what I do is I basically subtract the difference between the growth rate together and alone, and then that gives me the influence of one microbe on the other. So the influence of B on A is how much A's growth rate changes.
I see that you report in species level, in strain level.
Yes, because the genomes get down to that level. So it's really, that's the name of the genome that we have. And there's, of course, that's the best we can pick.
So you take just the best strain in this,
Mm-hmm.
in this genome.
Based on the 16S. Of course, there's not really that much precision, but sorry. I'll try to hurry up a little bit here. Okay. So you can sum up these scores. You can get your influence scores. And and then you can ask how important is each node. You can visualize it. And then you can start asking questions about the ecology and ask who are the different ecological drivers. And so just as a quick example, Peptostreptococcus, which is this P. stomatis down here, is very commonly associated with colorectal cancer. And you can see it's not, however, an ecological driver. It's really slaved off of other organisms in terms of its reliance on the environment. And you can look through for other insights like that. And so a couple of the really interesting things is that fuso doesn't play an ecological role. And a lot of people, I think we would also hypothesize that it's that it's probably interacting with the tumor, but it's not sort of driving the ecology. So it's not necessarily a driver. And that there are a lot of, and you can see who are the real players in the ecosystem, and that's usually primary fermenters and, interestingly, opportunistic pathogens. And you can do that for pMMR and dMMR. And I'm not going to go over it too much, except to say we have this paper, which I'll leave up. And I need to thank a lot of people for the research. And that's it. Sorry I went over time. Oh, well, yeah. I had 30 slides, and I thought that would be OK. But
so why don't we take one question, and then we have
nobody wants to be that one?
the validation, the level of complexity of these models is amazing, impressive, inspiring. But with so much going on, properties that you're trying to predict and validate on.
Yeah, so we're doing a couple of things. One is we're hoping to validate in like a bioreactor model. So we sort of just hunt around for problems that we can validate on, right? So one is we're trying to work with collaborators to put together a more controlled bioreactor and then say, can we make a modification? Because if we identify keystone species, we should either by sort of a targeted knockout of that species or by adding a species that could potentially act as a keystone, flip over that whole thing. Right? So that's one. We haven't really done it yet. But then two, we have looked at some of my colleagues' C. difficile data. And based on models like these, we have had success predicting which patients are, or which mice in this case, are humanized, germ-free mice, are C. diff susceptible and which ones are C. diff resistant. And we can assess this by asking, well, how would C. diff interact with the rest of the ecology? Would it have a bunch of positive interactions with everyone who's there, or would it have a bunch of negative interactions with everyone who's there? And if it has a bunch of negative interactions, that mouse is C. diff resistant. So even if you introduce C. diff, it sort of clears out on its own. And if there are a bunch of positive interactions cooperating with C. diff, that mouse is C. diff susceptible. So we have these little glancing ways in which we try to validate this. And I would say the other thing we do is we assess other aspects out of these models, which we can take these models, for instance, and look at the elasticity of the metabolic model. So there's multiple solutions you can get out of the flux balance analysis, and we sort of say where are the sensitive points. Like which metabolites is that microbiome very sensitive to? And a lot of those metabolites that the ecosystem is sensitive to are cancer-associated metabolites when people look at colorectal cancer and the metabolomics.